Your brain is one of the most important organs in your body. It contains all of your thoughts, feelings, and memories, and it's what makes you who you are. It's pretty amazing. But unfortunately, we don't really understand how it works. We know about neurons, those squiggly tree-shaped cells that carry electrical impulses and send and receive signals through neurotransmitters, but we don't know how neurons form themselves into distinct networks in their brain and how each of these networks perform specific functions. Luckily, since the new millennium, we've had an important tool to help us figure these things out: optogenetics. Alright, so what is optogenetics and how does it work? Optogenetics is a technique of turning neurons on and off using light. Now the opto part makes sense, but what about the genetics part? Well, a regular neuron doesn't respond to light. We actually have to introduce a light-sensitive protein, and we do that by bringing in the gene encoding for it. Usually, a virus carrying the gene for the protein, altered so it doesn't cause disease, is injected into the target area of neurons and it introduces the gene into the neurons. Okay, now we know what optogenetics is, so let's discuss how it works. Firstly, without any light-sensitive protein, a neuron carries electrical impulses by controlling its balance of positive and negative charges with the help of non-light-sensitive protein channels. When a neuron is at rest, its inside is more negatively charged than its surroundings, which have positive sodium ions. When a neuron is excited, its protein channels open and let in sodium ions, triggering an action potential, which is an electrical signal that travels down the axon to the axon terminals. Traditionally, electrodes were used to excite neurons by sending electrical impulses, but this didn't allow for high specificity in which neurons were excited or in the duration of the stimulation. With optogenetics, there's much more precision in which neurons are excited, and since we have more control over light, there's more precision in the time of stimulation as well. Additionally, optogenetics provides a way to directly inhibit neurons, whereas electrodes are only suited for exciting them. There are actually two light-sensitive proteins used in optogenetics: channel rhodopsin and halorhodopsin. Channel rhodopsin responds to blue light and it lets in positive sodium ions, turning the neuron on. Halorhodopsin responds to yellow light and it lets in negative chlorine ions, turning the neuron off. This gives us a way to directly inhibit neurons. So how has optogenetics helped the field of neuroscience? I said before that neurons are squiggly tree-shaped cells, but neurons actually come in many different shapes and are classified into subtypes. There are hundreds of neuron subtypes, and each subtype could potentially act in a different way. Neurons also form themselves into circuits, or networks. With the use of optogenetics, neuroscientists have been studying the behaviors of different neuron subtypes within their neural networks. Thanks to optogenetics, neuroscientists will be able to gain a new level of understanding of not only how the brain works, but how the brain can fail to work, leading to diseases such as Alzheimer's and Parkinson's. As this knowledge is accumulated, we will be able to treat, prevent, and possibly even cure neurological diseases with increasing efficacy.